Okay, so we're going to continue on with B5A. B5A says you're going to describe the stages of the cell cycle, and that's what we were doing. We're going to hit DNA replication a little bit later on in the next unit, and mitosis. So describe the stages of the cell cycle and mitosis, and then talk about the importance of the cell cycle to the growth of organisms. So the importance of the cell cycle to the growth of organisms is the fact that this is how organisms grow. If you want to add cells to the body, it has to go through mitosis. The cells you add more cells. If you want to replace damaged or dead cells, you go through mitosis in order to do that. So that's the importance of the cell cycle to the growth of organisms. We also talked about recognize the significance of meiosis to sexual reproduction. Sexual reproduction, the whole purpose of sexual reproduction is to increase diversity. So when we talk about meiosis down here, it's all about increasing the diversity. So the offspring are not clones of the parents. We talked about the fact that both of these are normal cell division, where the DNA of the mother cell are copied and divided equally between here in mitosis it's 2 daughter cells, here in meiosis it's 4 daughter cells. In mitosis, you have clones. All the cells have the exact same DNA. In meiosis, you do not have clones. These cells end up with only half the number of chromosomes that they started with. For a human, that would be 23 chromosomes instead of 46 chromosomes. So in mitosis, they're just making clones. It's how you grow, how you replace dead cells. This is how you make sex cells. And remember, sex cells are also known as gametes, and there are 2 sex cells, the sperm and the egg. So here's one of your vocabulary words up here. If you want to circle that, if you want to do something like that, there are the sex cells up there. So I'm going to ask you define gametes and give me 2 examples, and those are sperm and egg here. As we go down through here, we start off with a cell cycle, and we have interphase. It means that time between. This is where the cell spends most of its lifetime. It's going to be an interphase. It's doing its little cell job. Towards the end of interphase is when the chromosomes when the DNA starts to get ready to prep up to reproduce so the cell can make copies of itself. So the DNA is in chromatin form during this time because it's working form. When it's all loose and spread out, here's another vocabulary word right here. When it's all loose and spread out, that's when you have the work being done of protein synthesis, the cell's doing its job. Towards the end of interphase, that's when the DNA is going to make a copy of itself. It's replicated. Also towards the end of interphase, the cell checks the DNA for damage. It's one of the checkpoints for your body to check and make sure, hey, is everything okay with the DNA as it should be? So we start off with interphase. The next phase of the mitosis is prophase. And prophase means, if you just look at the prefix prophase, it just means the time before the DNA is quite ready to go. So this is when the chromatin starts to condense, and instead of that nice plate of spaghetti that we talked about where it's all loose and tangled up and spread out, what you have here instead is it's starting to condense down so it can be moved around. So remember, we're going to condense it down into, and it says chromatid form. Chromatid is always 2 pieces. It's like the original chromosome and its copy hooked together. So they're called sister chromatids when they're hooked together like that. Okay, so I'm filming this, so ask quickly. Yep, they're in the container over there. Okay? Yeah, and I'm going to have a sub tomorrow, so you'll have you're doing some, crap, I don't know what you're doing. You're doing the AP, there's a review for AP College Board, and then I have 2 videos from the packets in there. They're 2 different crash course chemistry stuff on thermochemistry, on electrochemistry. Okay? We took some notes yesterday, so you might want to copy those down from someone. Okay? Right. How'd you guys do? Oh. Did you do well? Yeah, good. Good. See you guys later. Okay, so the DNA is going to condense down into this chromatid form, and so it's basically the original chromosome hooked up to its copy. So we call these chromatids or sister chromatids. So the original chromosome and its copy bound together. Now, the nucleus during this time is also going to break apart because we're going to break out all that DNA out of the nucleus. Remember, it's kept safe inside the nucleus? We're going to break it out. So the nucleus just disappears. The membrane just breaks up. And then these things called spindle fibers form, and we'll talk about the spindle fibers and the centrioles in just a little bit. Now, remember that the chromatid, it's the chromosome, one complete strand of the DNA condensed, and it's always going to be hooked up to its copy. Okay? the chromatid is going to be one strand condensed and it's always going to be hooked up. When we talk about chromosomes and having 46 chromosomes in a human, that's one strand. When we talk about just one strand of that DNA. Prophase over here is really similar. The chromatin's going to condense, but instead of just having the original chromosome and its copy, remember you have 2 of the number 1 chromosomes, you also have the other number 1 chromosome and its copy hooked up together. So you have 4 different strands of DNA hooked up together here. And while they're in this tetrad form, what happens is you have pieces of things that swap on the chromosomes. It further increases the diversity. That's the key right here. This is why meiosis even makes it more diverse, because you have this stuff called crossing over. Here's another really important vocabulary term that we'll talk about. So we have crossing over happening in this process. Okay, once you have this down, what I want you to do is I want you to flip to your next page and try and get your line that you've made here. Try and get your line about the same place. So what I did is I kind of held my paper up a little bit and I made a mark here. And then I used my straight edge when I turned the page. I'm going to use my straight edge and I made the line here. So now we're going to continue on. So take a minute, pause this if you need to. Take a minute and set up your paper. And then we're going to continue on and talk about the next phase. So we said interphase, prophase. The next phase is metaphase. Let me see what colors. I'm using blue and brown. So let me get the brown and the blue out of here. So blue for mitosis. Okay, so the next phase of mitosis, and remember mitosis is on this side, meiosis is on the other side. The next phase is metaphase, and I'm going to bring it all the way up to the top because I'm going to need some room here. So metaphase. Remember, we're going to put boxes around these, so all the phases of mitosis and meiosis have boxes around them. And on the other side, we have metaphase 1. If there's a metaphase 1, there's got to be a metaphase 2 in here somewhere also. So this is metaphase 1. Okay. During metaphase, meta, that prefix meta, means middle. So during metaphase, the chromatids line up in the middle of the cell between the centrioles. Okay, we talked about centrioles before when we talked about the cell parts. And really the only thing we talked about what the function of the centrioles is that they helped in cell division here. So when we look at this, I'm going to show you a picture here. This is page 19 again. Here are the centrioles. They look like 2 little barrels crisscrossed here. But basically what's going to happen is these centrioles are responsible for throwing out these little fibers. Look at this down here in the second picture. These centrioles send out these little bitty, they're called spindle fibers. And they hook to one side. Each one hooks to another side, and they're going to be the ones responsible for pulling them in. Again, I always think of them kind of like fishing lines. So this is my fishing pole. I'm going to throw out a line and hook it to one side, and on this side somebody else is going to throw out a line and hook it to the other side. And then they reel them in. They pull them to different sides. So this is how the chromatid and its copy, the chromosome and its copy here, is going to be separated into 2 separate cells here. So the centriole creates spindle fibers to reach out and grab hold of one side of this little chromosome and copy and pull it over here. Okay, so the centrioles, I'm going to say throw out, or make spindle fibers that hook to the chromosomes. I'm going to put chromatids here so I can keep it consistent and separate them. Okay, so that's what's going on in here. This takes a long time. This stage right here, it takes the longest time. And the reason it's going to happen, so bear with me here for a second. This all doesn't happen during metaphase. I'm just telling you what the centrioles do. Okay, so this whole separating them thing doesn't happen. What's happening here is that the chromatids line up in the center. Everything has to be just so. So when we're talking about metaphase, we're talking about the fact that these things, all these chromatids and their copies that are hooked together, line up neatly in the center of the cell so that everything is organized, and it makes sure that it's all set up so that we can pull them safely and carefully to each side of them. So this is going to take the longest time. Out of the prophase, metaphase, anaphase, telophase, this takes longest. So I'm going to put P, M, A, T. So out of these four parts, this one, metaphase, takes the longest. You've got to line things up and organize them neatly for this to happen. In metaphase 1, instead of just talking about the chromatids and their sisters that line up, we have the tetrads. Remember, we've hooked them together in groups of 4 in the meiosis. The tetrads line up between the centrioles. And then it's the same thing is going on here. So this piece right here, I want you to be careful. This piece right here is just a definition. It doesn't really happen during the metaphase. I just put the definition here, so don't let that get confused in here. So I'm going to put it in kind of in little dots here in this case. And that's all metaphase is. It's the point where we're going to get everything organized. Everything's going to line up in the middle of the cell so we can get things organized and set out. The next piece would be anaphase. And over here in meiosis, it's anaphase 1. Okay, so during anaphase, that prefix ana means apart. So what do you think is going to happen to the sister chromatids? You've got the chromosome original and its copy hooked together. What's going to happen to them if the word means apart? Well, what's going to happen is that the chromatids are pulled apart to either side of the cell and are now called chromosomes. The sister chromatids, so remember, a chromatid is you've got a chromosome and its copy, and when they're hooked together, they're called chromatids. So the chromatids are pulled apart to either side of the cell. How about if I put sister? Let me say sister chromatids. If you guys put the word sister in there, it makes me happier. Sister chromatids are pulled apart to either, to E-I-T-H, either side of the cell. And this is by the centrioles and spindle fibers, right? Remember, those are the things that are going to pull them apart, that are going to separate them, okay? And once they're pulled apart, they're called chromosomes. After they're separated, they're called chromosomes. So yeah, they call the DNA lots of different names. So when it's loose and free and doing its job inside during interphase, it's called chromatin. When it's condensed and replicated, so you have the original and its copy hooked together, we call them sister chromatids. And then once you pull them, separate them apart, now you have individual chromosomes here. This happens pretty quickly. Now, over here on anaphase, we don't just have them in a group of 2, where we have sister chromatids together. Here in anaphase, the tetrads are pulled apart and are now called chromatids. They're now in the form of sister chromatids. So you've got the original plus the copy. So remember, we started out with a tetrad. We have the original and its copy. We have the other one and its copy. So the 2 number 1 chromosomes and their copies hooked together, and then they hook all those together. So you have 4 of them hooked together. Now we've just pulled them apart. So here's one of the number 1s, and here's the other of the number 1s. But because they were together, it's more organized so they can separate them out better, make sure everything has what they're supposed to have, and it allows for that crossing over where you can have pieces of things swap out and switch. And then the rest of the stuff is going to be the same. The last phase for mitosis anyway, the last phase is telophase. Okay, and if we have telophase over here, we have telophase 1 over on this side. Alright, now in telophase, the first thing that happens is cytokinesis. K-I-N-E-S-I-S. Cytokinesis begins. Okay, here's another vocab word, cytokinesis. Cytokinesis means cell, C-Y-T-O means cell, kinesis movement, cell splitting is what we're talking about. This is when the rest of the cell splits apart. So it's all been about the DNA, making sure you have copies of the DNA and splitting it apart. Now this is where you're talking about the rest of the cell splitting. So now you're splitting up between two new cells, you're splitting up the cytoplasm and all the organelles, so everything's kind of splitting in half. So each of the new cells has part of the old original cell that was there. So we have cytokinesis begins. That's the cell splits into two new cells. Okay, the nucleus in this stage begins to reform. So we got rid of it earlier on in prophase. Now we're going to reform it again. The chromosomes convert to chromatin form. So after it's all split apart, the chromosomes are moving back out. So the chromosomes convert to chromatin form. I can't write this. Let's try this again. The chromosomes, too many C's in here. The chromosomes convert, so they change to chromatin. It means they're loose and spread out. So once they've reformed their nucleus, they're going to spread that all that DNA is going to loosen back up. And if we're talking human, that's 46 chromosomes, right? And then here the cell goes into interphase. We're done. Okay, so that's what's happening here. Now, what I'm going to do here is tell you a couple of other pieces of information. Each cell is diploid. How many is di or bi? That means 2. So when we say diploid, it means that you have 2 of each chromosome, which is exactly what we started out with. So diploid means 2 of each chromosome, exactly like the parents. They're clones. Like the parent cell, the mother cell. Okay? So for humans, it would be 46 chromosomes, and they're 2 of each one. That's what diploid means. So 2 of the number 1s, 2 of the number 2s. I keep saying that over and over again, but that's what we're talking about here. Now, if it's not a human, it's going to have a different number of chromosomes, so you'd have to just look them all up. Over here in telophase 1 of meiosis, you've got basically you've got cytokinesis beginning. So same about cytokinesis. You have 2 cells that are forming. 2 cells with 46 chromosomes. And the cells don't stop there. Things aren't over here. The cells prep to go through another division. Because again, we don't want 2 cells. We don't want the original and its copy. What we want are half of that. We want either the original or the copy in one cell. The cells they prepare themselves to go through another division. That's what they're going to be doing. So now we're not going to go back into interphase. That would mean we stop and we start making things doing the cell job. So we started off with interphase, prophase, metaphase, anaphase, telophase. Now we're going to hit the prophase, metaphase, anaphase, telophase again. So here's prophase 2. In prophase 2, the spindle fibers attach to the chromatids. They actually kind of help in the lining up of when the chromosomes and stuff line up in the center of the cell to get ready to split. The spindle fibers help to do that too. So they attach to the chromatids because they just unattached because they brought them over to either sides. Then we have metaphase 2. Second time around is much simpler and more straightforward, right? The thing that took the most time is this little metaphase, lining them up in the middle. Takes a little bit less time this time around because things are pretty well organized for the most part. Metaphase 2, the chromatids line up in the middle of the cell. Remember, it's the original and the copy. The chromatids line up in the center of the cell. Anaphase 2. What's going to happen to the chromosomes here? You've got the original and its copy, so now we're going to pull them apart. So the chromatids are pulled apart and they're now called chromosomes. They're separated, pulled apart, separated. So you have the original hooked up to its copy, so they're separated, and they're now called chromosomes. Right? And then we have telophase 2. Okay. Oh. Now we have telophase 2. I've got to move this down so you guys can see better. So now we have telophase 2. And in telophase 2, the next stage, cytokinesis, begins. This is the final cytokinesis, so we're going to split all the parts, all the rest of the cell juice and all the cytoplasm and stuff. We're going to split those. We have the nucleus reforms. And if you look in meiosis, the nucleus never reformed here because we weren't finished yet up here. If you look at mitosis, we've got, the nucleus begins to reform. But here in telophase, we are not finished. We have to go through another cell division. So now, once it's gone through the second part of the cell division, now you can reform the nucleus. A total of 4 cells are made. Each with half the number of chromosomes as the parent cell. And that's called haploid. Okay, so diploid means 2 of each chromosome. So here is a vocabulary word. Haploid means you have half the number of chromosomes as the parents, so you only have 1 of each of these. Okay? And that means if we're human, that's 23 chromosomes. And this is the end. It doesn't go into interphase. Not yet. These are weird. These are sex cells. They're totally, totally different than other cells. a lot of times they're in some type of stasis in many cases. So like with the female, the sex cells, she's born with all of her sex cells. All the eggs are already made inside the ovaries, and so they're in kind of a stasis. The eggs, they kind of are in a stasis there. With the male, the sperm, they're going to stay inside it. They're not doing their job. They're not making proteins or anything like that. As a matter of fact, notice nowhere in here did it talk about the chromatin, the chromosomes moving back into the chromatin form. So look right over here. In telophase, the chromosomes convert to chromatin form. They don't do that in the sex cells because remember, they only have half that's what they're. They can't function fully as a normal cell because they're not just normal cells. Remember, the sperm is really small, and all its job is now to do is to transport all those chromosomes, the half chromosomes that it has, into the egg whenever it gets there. That's its function. It's not going to make proteins. It's not doing anything like that. It's just going to transport all those chromosomes. And the egg doesn't have its full complement of chromosomes. It can't carry out anything either. So they're not going to go into interphase again. Over here in meiosis, you're just making normal body cells, and so when it gets to telophase, it's going to kick in and do the rest of the stuff, and ultimately, this one's going to go back into interphase. So right here goes back to interphase. Okay? Now, I have one more thing, and I don't think I have enough space down here to draw this, so I might put it on a separate page, but we'll do that on the next day of notes, okay? So make sure your notes are in order, and then we'll finish taking these notes